Thanks for joining, everyone. This is Megan Weber. I'm an aquatic invasive species extension educator at the University of Minnesota, and I'm excited to have you join us all for the next in our aquatic invasive species webinar series. We have a guest who, if you've been watching these for a few years, you've maybe seen before, but we have Dr. Mike Smanski here to share more about genetic biocontrol, focusing on invasive carp species. I have a couple of housekeeping items, and then I'm going to pass everything over to Mike. He'll use about half of the time for presentation, and then after that, we'll have lots of time for Q&A. So save up your questions. We're going to use a tool called Slido for moderating the Q&A. I will put the information for how to join into the chat, but then also once we get to that portion, I'll put a QR code up on the screen for you to join in. We are recording today, so you can find our recording on our YouTube channel. You will get an email once the recording is ready. It takes us a little bit of time to get that transcribed. Everyone is muted for the duration of the webinar, so if you do have questions, go ahead and use that Slido tool. And if you're having any technical issues, you can use the chat. And we have Michelle here behind the AIS Detectors program screen name, and she can do her best to help you with whatever technical issues might be coming up. I think that's all that I have for you in terms of housekeeping. So I'm going to turn it over to Mike so we can learn all about the latest in genetic biocontrol.
Great, thank you. So, welcome everybody. I'm gonna make Megan a little bit of a liar and probably take a bit over half of the time to go through some slides. But um, last time I went through the slide deck, it took me half an hour and I added a few more for context. So I changed the title here. This is going to be a talk about genetic options for controlling carp. But I wanted to put it into some more context and I wanted to make you think a little bit more. This is not going to be just like a teaching webinar where I'm telling you about the options that are out there. This is going to be like a frontline journey through some of our research that MASERC has funded over the past few years. And the first thing I wanted you to think about is this term invasive species or aquatic invasive species. We're going to talk about aquatic invasive species today, so we'll be in the water talking about invasive organisms from elsewhere. But really, this last term, species, what does it mean to be a species? I'm going to kind of challenge your conceptions of what a species is in the talk today, because you'll see we're going to be breaking species barriers and making new species barriers uh, using genetic engineering in the lab. Here are some big numbers about species. It's estimated that somewhere between 5 and 50 billion species have existed on Earth. 99% of those have gone extinct. Um, Insects are the most speciest type of organism. There's over 1 million named species of insects today. What surprised me when I started learning more about scientific definitions of species is how diverse the definitions of species as a concept is. The definition that I'm going to use today is probably the version that you would have learned in high school. It has to do with mating barriers. So if there's two groups of animals and they can interbreed and have fertile offspring, they're considered to be the same species by the biological species concept. That's one of the 26 definitions. But if you have two groups of animals and when they interbreed, their offspring are either inviable or infertile, then they're considered different species. This is the example that you may have heard about with like horses and donkeys. They're different species, even though they can interbreed to make mules. So there's two major types of speciation events, right? With those 50 billion species that have existed on Earth, they come from somewhere, and there's different ways in which new species groups can be made. So one type of event is called a prezygotic barrier. If there's something that prevents the sperm from one type from ever seeing the eggs from a different type, that's called a prezygotic barrier. It can be physical, like a uh, uh, some kind of difference in morphology that prevents two groups from mating. It can be geographical, like a mountain range rising up between two subpopulations that would prevent their genes from mixing. The second type of speciation event is called a postzygotic barrier. This is if a sperm and egg meet, there can be genetic incompatibilities between the two types that prevent that zygote or that hybrid from surviving. And there's a mechanism that was posed by a famous evolutionary biologist named Smanski. Now this is called the Smanski-Muller incompatibility hypothesis, where he says if there's an ancestral type of organism and that diverges into two different lineages, random mutations can accumulate in those two lineages. And sometimes those mutations that occur separately in lineage A versus lineage B will be incompatible to each other, so that if those two mutations existed in a single cell, that cell couldn't survive. And so he postulated that this is one way in which these genetic incompatibilities can arise. And in my lab, almost 10 years ago now, we invented a way to do this. And I'm going to show you the first proof of concept before I tell you how it works. And I'm going to click these videos. We chose to demonstrate it as a proof of concept in yeast because yeast is the simplest sexually reproducing organism we could think to work on. And initially, we engineered a type of yeast that I'm coloring green here. And when we mated it to wild type, we saw no colonies growing on the plate. And so we looked at what was happening under a microscope. And those hybrids that were forming between our engineered species and wild type are shown at the bottom. You can see they're swelling up like water balloons. Uh, and they would almost burst in synchrony, like it's the 4th of July. And compare that to what a normal yeast dividing and growing should look like, which was in the video on top. And so this was great. This is the first proof of concept that we could engineer genetic incompatibility in yeast. And we did it with a new class of tools for controlling gene expression. We weren't putting toxins into the hybrids. We were merely turning on certain genes to levels that were lethal. In yeast, we were turning on a cytoskeletal gene that was making the cells blow up like water balloons. We're doing it with a set of tools that are based on CRISPR-Cas. You may have heard of CRISPR-Cas. It's an emerging tool for genome editing. We're not actually using it for genome editing here. We're using a version of Cas9, which is normally the molecular scissors that cut DNA. We're using a version of that enzyme in which we've mutated the two parts of the enzyme that are responsible for cutting DNA. So our version of Cas9 is called dCas9 for dead Cas9 or deactivated Cas9. It can't actually cut DNA, but we can still direct it to go anywhere we want to in an organism's genome. And to that dead Cas9, we'll fuse other proteins that are responsible for turning on genes. It's a process in biology we call transcription. So that means that we can develop a tool that will turn on any gene that's already in an organism cell to stronger levels. Now, it turns out I told you in yeast we were turning on a gene that is responsible for the cytoskeleton. We made the cells blow up too big and their membranes couldn't contain them. But in other organisms, you could imagine in development turning on a gene that's essential for heart development. If you turn that on in the brain's not going to develop properly, or vice versa. So every organism has these genes that have to be turned on in a certain time or a certain place during development. And if you turn them on too strongly, it's bad for that organism. So we can create what we call a synthetic species or an engineered genetically incompatible organism that has one of these dead Cas9 gene activators that's looking for a gene to turn on to lethal levels. But in that engineered organism, we've introduced a small 1 or 2 base pair mutation. Let me make sure that my pointer is activated here so you can see, I can direct your eyes to some parts of the slide. So we can introduce a tiny change to the genome of our synthetic species that makes it immune to that programmable gene activator. In essence, you can think of the gene activator like a sentinel that's searching for the natural or wild version of a DNA sequence, but it never finds that version in its own cell. So the synthetic species has no ill effect from carrying this activator. It can reproduce with similarly engineered organisms, have offspring, and they're happy and healthy. But if it ever mates with a wild version of its type, then that sentinel will find the natural DNA sequence and drive that hybrid lethality. Now, we're excited with this as a paradigm because there's a lot of genes in any genome that are lethal when they're turned on too strongly. So we think that we could use this to create not just a single engineered species, but an arbitrarily large number of synthetic species that are all incompatible with the wild version and incompatible with each other as well. Now, some of the applications for this would be if you want to make a GMO crop and contain the transgene so that it can't jump out into wild plants or food crops, you could make it a synthetic species. In the world of pest control, I'm showing here an example in insects. I'm going to show you in the next few slides a proof of concept in insects. If you made, for example, a synthetic species in mosquito and released only the males, any female that they mate with won't have viable offspring, right? You can think of the male's sperm like a pesticide, except unlike chemical pesticides, it's very species specific because the male insects that you release aren't going to try to mate with butterflies or honeybees. They're going to only mate with the females of their same type. And so it's very precision pest control option. And it's one of many types of genetic pest control options that have been developed in the last decade or so. And so when we had that proof of concept in yeast, a lot of reviewers of papers and proposals told us it was a neat parlor trick, but it would never work in a more complex, higher eukaryote. So we demonstrated it in insects. And I'm going to gloss over many years of work to show you this video. And actually, it looks like I'm going to have to get rid of my laser pointer to play the video here. On the left is a time-lapse video that was recorded over 15 days where after we had engineered the synthetic species, we mixed three males and three females. Those are the adults that you saw at the start of the video. And then after 3 days, we took them out. So they didn't just die. We took them out of the tubes because really what we're interested in seeing is what happened with the eggs that those females laid in the media at the bottom of the tubes. And you can kind of see the punchline already. So on the leftmost tube, we had an engineered genetic incompatibility, EGI, engineered genetic incompatible, male and female. In the right, we had wild type male and female. And in the center tubes, we had either a female EGI and a wild type male or vice versa. And what you can see, well, we had a few larvae and pupa form in the middle tubes. None of those survived to adulthood. We saw 0 adult surviving progeny. So this was the first data that showed we could actually engineer the speciation event in a higher eukaryote, an insect. Now we used Drosophila melanogaster, which is a model lab insect, not a invasive species or an aquatic invasive species. But I'll get to that in a second. Further, we showed in that paper that was published 6 years ago that we could do what I claimed and engineer many different genotypes of synthetic species for Drosophila melanogaster. Those words at the top, hedgehog, pyramus, and wingless, are different genes that we were targeting for overexpression or turning on too strongly. And we had several different genotypes where when we mated the males to the females, we could be incompatible with wild type, which in this case is Oregon R. That's the name of the wild type melanogaster line, and incompatible with other genotypes of synthetic species, but they would only produce offspring. That's shown on the diagonal here. The colors correspond to how many surviving adult offspring there were when we mated males of the top genotype to females of the genotype listed on the side. And you see we only saw surviving adults when we were mating the same type with each other, but never when we mixed types. Okay, we since have gone on to publish a few more papers in insects showing that this works in cage trials for pest control, but I want to skip over that, and I'm going really quickly because I want to get to the aquatic work, which is why you all are here. So moving from a model species like Drosophila melanogaster to an applied pest, whether it's an insect pest or a fish pest like an invasive species, is challenging. We don't have the same genetic tools that have accumulated over decades that we have for the model laboratory organisms. Rearing can be more difficult, things like that. And so this story is really going to be about how we're translating this promising new technology for pest control into common carp and eventually other aquatic invasive species. Now, the first thing that we wanted to confirm is what genes can we target in fish that will be lethal when they're turned on too strongly. In this case, we used zebrafish, thinking that the genes in zebrafish are similar enough to other invasive fish species that a gene that's lethal when turned on too strongly in zebrafish, a model lab fish, should be lethal in a common carp or black carp or a grass carp. So we tested about a half dozen different genes, and we found three, shown on the left, that had zero embryo survival after 3 days. This is, for example, what happens if you turn on a gene called GATA5 too strongly on the bottom right here. Let me get my pointer back on. So after 1 day of overexpressing the gene GATA5, the embryos are all dead. Next, we had to find a version. This is a complicated slide, but this is a slide showing that we're able to find versions of that dead Cas9 activator that was strong enough to turn on GATA5 to lethal levels. The key thing to look at here is this box that I'm highlighting with my pointer now. That's showing over the first 3 days when we're turning on GATA5 with a dead Cas9 that was directed with something called a guide RNA number 3. We saw zero surviving larvae, zero surviving embryos, sorry. And compare that to controls where we saw good levels of survival. That convinces us that we have the genetic parts that we can use to engineer this genetic incompatibility in fish. Next, we had to develop methods to actually engineer an aquatic invasive species like common carp. The first few years of us trying this, we would take a boat, go out roughly this time of year, try to catch a carp female that was gravid, that was holding eggs, rush her back to the lab, get a male, rush him back to the lab, and then express the sperm and eggs, do in vitro fertilization in a plate, and we would get a chance to try to introduce foreign DNA to that fresh embryo to see if we could genetically engineer that carp. And if it didn't work, we would try again the next week, and if it didn't work, we would try it again the next week. But carp only mate in this part of the country over a few weeks every year. So if we didn't get a successful transgenic in those weeks, we would have to wait till the next year to take another shot on goal. And that just wasn't compatible with the level of throughput that we needed to engineer a complex system into a species like common carp. So we developed a lab-based spawning protocol where basically now we have cohorts of fish that we move between tanks of different temperatures with different daylight cycles so that no matter what time of year, there are some fish in our facility that are trained to think that it's spawning season. And actually recently we've been doing this every week now. We're able to take some of those fish and induce them to produce eggs and sperm. And so we're able to successfully spawn fish throughout the year. And we've refined this protocol to the point that we have a fairly high efficiency of generating genetically modified carp all throughout the year. Now, these pictures that you're seeing here are carp that have been modified with a green fluorescent protein from jellyfish. There's not really a function of that green fluorescent protein besides being a marker. The genes that we introduce that I'm going to tell you about for genetic pest control are invisible, and it's difficult to see which genes or which fish actually got that foreign DNA and which didn't. So we often will put right next to the genes that are doing all of the activity, we'll put right next to that a green fluorescent protein or a red fluorescent protein just to make it easier to pick those needles out of a haystack. Now, in animal engineering, we harvest these early embryos from in vitro fertilization. We'll inject DNA that we've designed and built in the lab into those embryos, but not every cell in that first transgenic fish gets that synthetic DNA. So the first generation is what we call a mosaic or an F0 transgenic fish. And that's where you get these speckly patterns. Some of the cells in that fish, some of the tissues get the transgenic DNA, others don't. But we really hope that some of the germline tissue, so the tissue that's going to become the sperm and eggs, is modified so that when we cross that mosaic fish to another fish in the next generation, we'll have some fish that are completely transgenic. We know that our success of modifying the germline ranges between single-digit percentages up to, in rare cases, we'll get 100% germline transmission. So, so we developed that ability to routinely make genetically engineered carp, and we realized that, you know what, there's been about a dozen or more different approaches for genetic pest control that have been demonstrated in model lab organisms in the past decade. but we're one of the only groups in the world that has this ability to genetically modify carp year round like we do. So with carp, we decided not to stay in our swim lane. With the insects, we're kind of continuing down with the method of pest control that we invented in the lab. But with common carp, we're kind of throwing the whole kitchen sink and trying out a bunch of different approaches for genetic pest control. Now, one of the dimensions on which genetic pest control methods differ is how long they will persist if released to an environment. Remember, the model for how these would work is you would release an engineered male into a lake if it's a common carp control agent, and that male will spread deleterious genes into the population. Now, some, like our genetically incompatible males, have no way to reproduce themselves. So they will fertilize eggs, those eggs won't develop, but the male won't be able to make another GMO offspring that will survive and persist. Some genetic pest control will persist in the environment. For example, there is a method uh, called female lethality that you can introduce a genetically engineered male that will skew the ratio of males to females in the population. Now, it's more complex than that. Here I'm showing you five different kind of inheritance schemes for five different sex ratio biasing approaches that all differ in how many offspring will be had, how many offspring will be transgenic. Will the females survive? Will the females die? Sometimes the females will survive, but they'll be phenotypic males. And so there's an incredible amount of diversity of genetic biocontrol options that are out there. And this is just a sampling of that. Again, the goal of my talk isn't to give you detailed descriptions of all the dozens of genetic pest control options that are coming to bear, because an important step is for any particular invasive species, we need to see which methods can we actually get to work in the lab environment first. And I don't think, originally when we were throwing the kitchen sink at common carp, we thought we wanted to find what's the best genetic pest control method for carp. I've matured from that point. I don't think there is going to be a single best method, but instead there's likely going to be a best method for a certain body of water. If you look at the bodies of water where common carp are a problem, I'm giving three examples here. Utah Lake is a big reservoir out west, the Murray-Darling River Basin in Australia, and Long Lake in Metro Twin Cities. These are very different bodies of water. They differ in their size, their interconnectedness, how many spawning grounds there are from that body of water. And so it's likely that there's going to be a different solution. And some of these bodies of water, maybe genetic pest control won't be an appropriate part of an integrated pest management plan. But it could be that a sex ratio biasing technique will be better for one body of water, and a sterile male technique will be better for another. Potentially some bodies of water, you would need something as strong as a gene drive, which we're not currently developing, but that would be the most powerful type of genetic pest control. And so an important part of the work that we've done has been engaging with key stakeholders throughout. Um, we published this paper last year, developing a language based on the technology readiness levels, which is a framework for measuring the maturity of a new technology developed by NASA in the 1970s. We worked with regulators, federal and local, tribal representatives, watershed district managers to come up with kind of a roadmap for what would the development of genetic pest control for carp look like. Right now we're still at kind of TRL3, the proof of concept in a model organism. We have some fish in the lab that are potentially proof of concept in common carp. We're still in the process of characterizing them. But you can imagine a slow, safe maturity of that towards field readiness would include a test first in a secure pond where, excuse me, maybe an outdoor pond that would be able to test diverse environmental conditions, but that pond would be contained in a way that the fish couldn't get out to other bodies of water. You would also then progress to a larger field trial. And if the technology shows that it's effective and safe in those increasing levels of readiness, eventually it would be ready for deployment around the state or around the country or around the world to eradicate an invasive species. And again, with the non-persistent genetic pest control, because the males don't persist and spread transgenes to their offspring, you have your ability to take your foot off the gas at any point. So there is a measure of safety and redundancy built into it that if something goes awry, you can always kind of take your foot off the gas. But I wanted to discuss from that work that we did the a key dimension that one has to consider when thinking about bringing a new type of technology uh, out into the world is the social perception, the public perception, and also regulatory perception or regulatory approval processes. So we know that from doing surveys and talking to a lot of people, there's a general comfort with genetic pest control, particularly the types of genetic pest control that don't include genetic engineering. In insects, there's been genetic pest control that's been used to eradicate pests from the whole continent of North America. Although, if you read the news, you'll see that that pest, the New World screwworm, is coming back into North America. So there's a lot of renewed attention to it. But you can sterilize males using radiation. So it doesn't involve genetic engineering. You use radiation to sterilize males and you dump them out of planes by the millions. And the females that they mate with don't have offspring. In the world of aquatic invasive species, there's a type of genetic pest control that has been deployed out west called the Trojan male approach, where males are engineered to have two Y chromosomes, so they can only have male offspring. And this is a type of non-GMO sex ratio biasing. The goal is to skew the ratio of males to females in the population to the point that there's too few females to support a large population. Genetically modified pest control is possible, but is likely going to have more stringent regulatory approval processes and a tougher road to public acceptance. And so I wanted to introduce that, and I'm going to come back to a little bit more of the technical aspect of engineering fish in a lab, because I'm going to introduce you to a surprising finding that we had that's going to put a new dot on that map of genetic pest control options and how they relate to each other in terms of regulatory and public perception. So I glossed over this before, but I'd mentioned that these mosaic fish, we have to wait till they're sexually mature and mate them with another fish to see if we had engineered the germline, the sperm and eggs, so that we could pass those transgenes on to the next generation. In common carp, this is about a 2-year process. I often get the question of, when are you guys going to start working on grass carp or black carp or bighead carp? We would love to work on all of those, but there's a huge space and time requirement to work on a new type of aquatic invasive species. This pictured on right here is about a third the size of the carp rearing space that we have in the MISERC containment lab. And common carp requiring 2 years to reach sexual maturity is actually pretty good when it comes to invasive species. Others, like black carp, require 5 years to reach sexual maturity. So if we were doing the same technique in black carp, it would be probably a multi-decade process to do the breeding that we need to get to the final genotype of the biocontrol agent. So we had an idea to test a technique that had been developed in the aquaculture world. Now, this is going to sound like science fiction. It did when I first heard about it. We didn't invent the technique, but we're borrowing it from aquaculture groups, and it's called surrogate host technique. It turns out you can take two different species, one we're going to call the surrogate host species, and one is the donor species. To the surrogate host species, if you introduce, you can either do this with use of chemicals or some genetic approaches. You sterilize the surrogate host so that it can't develop its own sperm and eggs. And then to that sterilized host, you can introduce the stem cells from a donor species, and with some likelihood, with some frequency, those will engraft in the surrogate host, and you have a surrogate host species that produces eggs and sperm, eggs or sperm, of a different species. So our thought was, what if we took common carp and implanted their stem cells that will develop into sperm and eggs into a sterilized fathead minnow. One, fathead minnows reach sexual maturity in a few months, so it would dramatically accelerate our engineering options, our engineering process. And second, fathead minnows take up much smaller tanks, so we could reduce the footprint needed. Essentially, we're using fathead minnows as a petri dish in which we can rear the carp germline. And if you mate two of these fathead minnows together, you would produce a wild type common carp. And so this is a work in progress still in the lab, but we've made great strides towards getting it to work. So we have ways that we can label the stem cells that become sperm and eggs. Here we're labeling with a red fluorescent protein those cells in a common carp embryo. We have ways that we can sterilize fathead minnows. We're using a type of chemical called a morpholino that mimics nucleic acid or DNA. And when we dose these fathead minnows, here's a probe that will label the primordial germ cells, which are the stem cells becoming sperm and eggs. Here we're labeling those with green fluorescent protein. And if we introduce the morpholino, those cells don't develop. The fish still develop and are fairly healthy, but we can get near 100% sterility if we dose them with just 6 nanograms of this morpholino. Apologies to any of you that were eating lunch during this webinar. I should have blurred this image a little bit, but here's a dissection image showing the gametes of a sexually mature fathead minnow, a female on the left, male in the middle, and one of these sterilized males on the right. And we see a high degree of sterility. It's not perfect. There's a single-digit percentage of fish that don't become sterile. One thing that we were surprised to see is the fish that got the largest dosage all turned out to be sterile males. So it turns out if you sterilize a fathead minnow, it becomes a phenotypic male, even if it was genetically programmed to become a female. We think this is something that we could overcome to produce both sterile males and females. But we saw a surprise when we were testing this. And actually, I'm going to jump past that slide really quick. So we had a bunch of surrogate fathead minnow hosts to which we had introduced carp stem cells. And to test if it worked, we wanted to do a procreation assay. So the idea was let these minnows grow up for a few months. And when they're adults, let's harvest sperm from them. And the thought was if we try to fertilize a carp egg with the sperm that we get from the minnows, if it works and we get a living fish, then it means we really had a surrogate minnow that was harboring carp sperm. However, we knew that the sterilization of the minnows wasn't 100%. So there was, out of the 30 or so fish that we had to test, probably one or two of them were still just normal fathead minnows. They weren't surrogate hosts. So we thought we should probably test just to make sure that normal minnow sperm can't fertilize a carp egg. And that's where we were surprised. We actually made a new type of hybrid called a MARP. So the first part of this talk was about making new species barriers. We can make these engineered synthetic species. This is a part of the talk about breaking species barriers. This is a new type of hybrid that no one has described in the literature yet. Turns out a fathead minnow and a common carp can have a hybrid offspring. In the world of hybrid organisms, there's a naming convention. So you have to take the beginning of the name from the paternal parent and the second half of the name from the maternal parent. So that's where MARPs come from. Kinnos, which would have been the opposite, is actually not viable. So this is single directional. We can't use carp sperm to fertilize a minnow egg, but we can use minnow sperm to fertilize a carp egg. The MARP is shown in the top here. It has anatomical features of both carps and minnows. It has a forward-facing mouth, so it's unlikely to root around and uproot native vegetation the same way that a carp would. I'll show you some growth data in a bit. This is a slide to show that we repeated this shortly after with sperm from a fluorescently engineered minnow to confirm that the minnow's genome was actually turned on in this hybrid MARP. So in that case, the MARP glowed green. And we've done some characterization. So we've had MARPs in the lab now for almost 3 years. We know that they are somewhat intermediate between carp and minnows. They grow much larger than minnows, much smaller than carp. There is they're less viable than just wild type minnow offspring or carp offspring would be. So we see a significant lethality in the first few days, but those that survive past about 5 days have a good chance of surviving to adulthood. And interestingly, uh, they we predict that they're going to be sterile. Uh, those of you, I've learned more about mules in the past few years than I ever thought I would as a scientist. This wasn't my area of expertise. The reason mules are sterile is because horses and donkeys have a different number of chromosomes. One has 36, one has 38. So mules have 37, and that odd chromosome, that unpaired chromosome, makes the development of eggs and sperm fail in mules. Now, carp and minnows have crazy different numbers of chromosome pairs because in the evolution of carp, there's a genome duplication event at one point. So minnows have 25 pairs of chromosomes, carp have 50 pairs of chromosomes, and MARPs, we've done an analysis now and found that they have 75. So that's as expected. They're inheriting one set from the minnow parent, one set from the common carp parent. But because of that, we expect the MARPs to be completely sterile, and all the evidence that we have points to that. We've never seen a mature looking gonad in a MARP, and there's no evidence that they can have offspring with either carp or minnow parents. The interesting thing, though, and bringing this back to genetic pest control, is that we realized, this was kind of an example of life giving us lemons, and we think we can maybe make lemonade out of it. Originally, our goal with the surrogate host technique was to move carp germ cells into minnows just to make the logistics of engineering carp easier in the lab. And we're still moving forward with that. We still think that's a good route forward, especially to access some of the more difficult to work with aquatic invasive species. However, what if we go the opposite direction? What if we sterilize a carp embryo and inject the primordial germ cells or the stem cells that will generate minnow sperm and eggs? We could then produce an adult male carp that doesn't produce carp sperm but produces minnow sperm. And if that's released to the wild, any female that it mates with will have MARP offspring, which is an evolutionary dead end. Those MARPs, since they're sterile, they can't procreate. They would be eaten by fish in that body of water, but it would be a dead end for that lineage. So this could be a way that doesn't involve any genetic engineering that could, in theory, control the population of a fish like common carp. Um, so back to this chart, we call this approach surrogate males authoring sterile hybrids. I was trying to force an acronym here, and feel free to leave something in the comment section if you have a better acronym for it. But this idea doesn't involve genetic engineering. It's not natural by any means. It's still a hybrid organism, a surrogate host technique, but there's no transgenes involved. And so the regulatory approval process would be distinctly different for this type of genetic pest control compared to some of the ones that use GMO biocontrol agents. Releasing a surrogate male carp to a body of water wouldn't be releasing any genes that didn't already exist in that body of water. Fathead minnows are the most abundant fish east of the Mississippi River, and MARPs, well, an unnatural hybrid, are a hybrid that does form naturally if minnow sperm fertilize a carp egg. And so we're kind of exploring this now as an alternative approach to genetic pest control. And that brings me to the end of my slide deck. Looks like I was true to my word and went about 40 minutes. So I kind of asked you to think about this concept of species. In the first part of the talk, I told you how we have a way to engineer new species-like barriers to gene spread and how we can use that in a pest control application. And then the second part of the talk, I told you how we're breaking species barriers with novel types of hybrid organisms that could actually factor into genetic pest control. Uh, like most research, this leads to more questions than we had going into it. These are some questions that I commonly get, but I'll progress past this slide and get to your questions that you're leaving either in the chat or in Slido. But before I do want to acknowledge the we're really grateful for the support that we've gotten from MASERC over the past several years via the Environmental and Natural Resources Trust Fund. I'm picturing a few of the students that have done the work that I got to talk about today. This is a fairly old picture, but I liked it because four of the students were all standing next to each other in this one lab photo. But with that, I will, let's see, maybe I'll go back to this slide and, but I'm gonna stop talking and maybe Megan, do you want to, if you're controlling the Slido, do you want to ask the questions to me or is there a way I should go and find them?
Yeah, yep. I'll go ahead and moderate for you. Is it okay if I take this screen down for you to share the join code for folks for now? And then we can maybe
Yes,
pop this
yes,
one back up partway through?
I can
All
take it
right.
down myself.
Here we go. I'm gonna share this. So this is the link to get you into the Q&A. I know a couple of questions have been coming in already. So the way that this works, you can either use a mobile device to scan the QR code on your screen, and you can join the Q&A there, or you can type into the website, theslido.com, and enter the event code CARP to join in. And the third way, if Slido isn't working for you, if you put your question into chat, Michelle will help make sure it gets into Slido. The nice thing about Slido, if you join, even if you don't have a question, is you're able to upvote questions that you're particularly interested in. So you can also join in and make sure that the questions that you have are the ones that get answered. I'll go ahead and get started, but I'm glad you added those extra slides in, Mike. That was some great extra info. All right, here we go. So, first question of the day is asking if you know about how many modified carp would have to be released into a lake eventually to take care of the existing population.
Yeah, so we've done modeling to generate numbers, and it's highly dependent on the lake size, the amount of time that you have, the cost, the economics that you're willing to spend on it. But what we imagine is that these genetic pest control approaches are not going to be silver bullets, and it's not going to be the only approach you would use in a body of water. They're most effective when combined with other either physical or chemical approaches. So you can imagine, like right now, you can seine carp out of the bottom of a lake. They tend to aggregate in the basin at the bottom of a lake in the winter, and seining is a good way to reduce population numbers by 90%. The problem is that 10% that remains can reproduce quickly enough that they'll refill the lake again. So we think of genetic pest control with something like common carp more as like a mop up of those last few stragglers that that weren't controlled with the physical or chemical approach. You could even view, a sterile male almost like a prophylactic uh, type protection, where if you've eradicated the carp, or really with common carp, there's a known threshold below which they don't cause the same type of ecological damage. I didn't get into this, but Shemek Bayer has done great work in the past 10 years showing that below 100 kilograms per hectare, carp don't do the type of ecosystem damage. And so it's really about suppressing the populations below those numbers. But I think that depending on the genetic pest control technique and depending on your control goals, the key is that you're getting more of the genetically modified males in there than wild males. There are some exceptions to this. We know that in fish, large males tend to be more mating competitive than small males. So you could get by with lower numbers if you're releasing larger male control agents. A lot of variables, but yeah, we have run some models. I can't give you just a number though, because it's that number would be wrong in 99% of the applications. It depends too much on a specific application.
Sure. Great. Thank you. All right. Next question is about the MARPs and wondering how close you are to testing the MARPs in the wild.
Oh, so not close at all in the wild. Like I'd flashed up a graphic from our technology readiness level paper, and so I'm gonna walk the
Yeah.
walk that I'm proposing there and use the language that we proposed. So with MARPs right now, I would say we're still at technology readiness level 2 in terms of their application for genetic pest control. We've generated MARPs, we know they're sterile, but for them to be used as genetic pest control, the proof of concept that we need to do in a lab first is demonstrating that we can engineer these surrogate carp hosts, the carp males that are producing minnow sperm. We haven't gotten there yet. When we do, that'll be the proof of concept. So that'll be technology readiness level 4, actually. And we would characterize that in the lab to learn how effective is it? Are there any, risks that we didn't consider? And so we'll test the behavior of those males, the efficacy of those males. That's going to be a challenging technique to scale. But assuming we could scale it, the next stop would be kind of an external pond, maybe that's surrounded by fences, similar to what they have. There's a few of them at USGS facilities uh, that would allow us to test if the technology could work in diverse environments where it gets hot and cold every day. In our lab, it's pretty consistent temperature and lighting every day. And so we'd want to test it in a more environmentally relevant scenario. But it would be years and years before we did a wild release.
Great. All right. The next question might be another one. I'll ask it and you can let me know if you're just not quite there yet. But the question is around the cost and how expensive this mode of control can be for AIS managers, perhaps in comparison to some of the current control tools available.
The with any genetic pest control, there's more upfront cost in developing the genotypes and doing the engineering in the lab. But because, for example, with EGI, we would have a true breeding strain, the cost of implementing it would be similar to any stocking program, right? You would have a facility where you're rearing these and generating, thousands or millions of hatchlings and fingerlings. And so the cost would be on par with that.
Great. Okay. Next question on the list. Are the genetic engineering processes that you're describing considered gene drive? And I think you might have gotten to that towards the end a little bit too. But they're wondering about like how that those are different from each other and what risks that might carry.
Yeah. So gene drive is a type of genetic pest control that's self-sustaining. So when I talked about persistence, if you're releasing sterile males, they can mate with wild females to decrease the number of offspring that that female will have, but that sterile male doesn't have a way to make more genetic biocontrol agents. It's kind of a dead end. Gene drives are a way that you can engineer an organism that will self-propagate in the wild. And so there's a lot more caution. We're not developing gene drives in the lab. There are people that I know well, from other parts of the country or other parts of the world that are developing gene drives in different organisms. No gene drive has ever been released, even though genetic pest control, even GMO genetic biocontrol has been uh, done at field scale with certain insects, particularly mosquito varieties. And in South America, there's countries where it's gone commercial. There was a time you could buy genetically engineered mosquitoes at the supermarket in Brazil, and put them out in your backyard. But for a gene drive, that's never been released. And the challenge with the gene drive is that a field trial is essentially the same as the final application, because once you release it, that genotype can spread and suppress a population on its own. So, no, we're not working on gene drives per se. Those are a type of genetic pest control, but because they're self-spreading, it's kind of like the first binary question you ask of a genetic pest control technology is it self-spreading or is it not? Right now, we're only working on the ones that are not.
Great. All right. Moving on to the next question, wondering about other species. So have you or others looked at using these kinds of techniques with zebra or quagga mussels?
And I'll answer that, but Megan, I'll point you to there's a few questions in the Zoom chat as well that aren't in Slido. I see
Okay,
Paul has put them there, and I don't want those to get lost. So, so I would love. So to be honest and candid with you, um, my background is as a chemist. I'm a chemist that became a genetic engineer, And I was kind of naive when we were approached by people who care about aquatic invasive species, and they said, "Could you do this into common carp?" And we said, "Yeah, let's go for it." The amount of time and effort that it took to develop the protocols to work on carp, a non-model organism, was significant. And so I'm a little bit more guarded now with any new organism, because with carp, we really had to invent the wheel ourselves of how are we going to rear them in the lab? How are we going to do genetics? There's a student who graduated from my lab, Dr. Julie Badger, who's currently working with mussels and trying in a different lab to develop genetic engineering protocols for zebra mussels. And as soon as she's successful in developing the ability to rear zebra mussels through their whole life cycle and demonstrate genetic engineering is possible, then yes, there's many genetic biocontrol methods that I would love to try in mussels. And there's other biotechnology applications in mussels too, for example, in accumulating rare earth metals or remediating pollutants because of how good they are at filtering water. But the short answer is we're not working in mussels yet because there's not a reliable way to do genetic engineering in mussels. But I think that'll change in the next years.
Well, great. All right. I think I see now this next one, maybe two versions of this question in Slido, and this might get to one of the ones that's in Zoom. But this person's worrying about what your concept or definition is of when an invasive species becomes an accepted or common, or I think in another question, a naturalized species.
That's a great question. And to be honest, like answering it what gets me a little bit over my skis. Uh, the MACERC has a panel of experts that every year, every other year, meet to decide what are going to be the target species on our list of focal species that MACERC focuses on. And they consider that all the time. Now, I see your point. Uh, Over the course of history, species have moved from continent to continent. One thing I joke about is that like if we don't like invasive species, we kind of have to take tomatoes out of Italian food and potatoes out of Irish food and peppers out of Thai food. Those were all New World species of plants that found a home in other parts of the world. Yeah, I think included in the definition of invasive species is an amount of environmental or ecological damage they do when they're out of their range. And so it's well documented the damage that carp can do to aquatic ecosystems here in the States in decreasing the native flora, decreasing the abundance of native fishes, even changing a lake so much that the waterfowl no longer will go there. It impacts the lake's ability to sponge up pollutants that are running off from agricultural fields. So there's myriad reasons why this particular invasive species, common carp, makes sense to control, and that's why so much time and effort and money is put into common carp control. So I understand the point, and I understand that there's some opinion that we should naturalize carp at this point. Uh, but for now, I think there's good reason to still put effort in developing technologies to control carp populations.
That sounds great. I think I'm actually gonna skip. There's a couple that are above this, but the end of that answer, I think, starts to answer one of the other questions, which was effectively a question of like, is the war already lost on controlling invasive carps or common carps, or barkthorn was also here. And I think I heard you say that no, you feel like there's still opportunities.
So the uh, we're in the middle of a revolution in terms of genetic engineering right now. And just 20 years ago, when we first sequenced the human genome, that was a step change in biology. That was the ability to read DNA. And almost every area of biology was affected by it, not just human health and disease, but for example, once DNA sequencers were widely available, even people like lake ecologists could go and just harvest a liter of lake water and sequence all the DNA in it to know everything that was in that body of water. The paradigm shift that we're going through now has to do not with reading DNA, but writing DNA. So every year it's getting cheaper and cheaper and cheaper to synthesize DNA. The students in my lab daily will go to a website, type in a series of A's, T's, C's, and G's, which is the letter of DNA, and they'll get a day later a test tube that has that actual DNA molecule, and they can use that to reprogram a fish or a plant or an insect. And so the types of technologies that are going to be possible in the next 10, 20 years using genetic engineering are are amazing, I think. And so I hear you. This is a widespread problem. Common carp are on every continent now except for Antarctica. They're in really complex waterways. But these new class of technologies in genetic biocontrol are just in their infancy. And I think as these mature, things that seemed impossible previously are going to be possible in the future.
Great. I love hearing messages of hope. All right, I'll go on to our next question. This one says, when using the surrogate fatheads, what's the trade-off between the number of eggs produced due to the smaller adults versus the length of time it takes for a carp to reach maturity?
So when we're doing genetic engineering, the numbers of potential offspring that a fish has in our lab is not really important to us. You know, these fish produce thousands of eggs in each spawn, and we're happy if we get a few to be engineered. So that's not a concern of ours. The fact that the surrogate host will have less eggs per spawn, uh, we really just need access to, we don't even inject most of them. When we're doing the microinjection to introduce DNA, we might introduce DNA to a few hundred, but nowhere close to the thousands that we get from a mature adult female.
Okay, and then this person is thinking back to that question about zebra and quagga mussels, and they're wondering about goldfish since they're a little bit more closely related and what the opportunities might be there.
Uh, yeah, so interestingly, we've kind of, goldfish is another fish that's known to form hybrids with common carp. So a lot of the uh, technologies I introduced you to could, in theory, work for goldfish. Goldfish, they're not quite, while they're smaller than common carp, they have a similar age to sexual maturity, so they can be a challenge to work on in the lab from an engineering point of view. But yeah, so the challenge now is when I give talks like this, it seems like a lot of people in the audience have their favorite target aquatic invasive species, and they say, well, when is this gonna be ready for this, for grass carp or for black carp or for goldfish? One of the reasons we're really excited about the surrogate host technique is that it would allow us to start targeting many species in parallel. We don't have the space in MCL to have a huge footprint to, a dozen tanks to work on black carp and another dozen tanks to work on grass carp. But if we establish the surrogate host technique, the fathead minnows just take really small tanks, and we could have a rack where one shelf is us, en route to engineering a black carp biocontrol agent. Another shelf is fathead minnows that are bearing engineered grass carp. Another shelf might be fathead minnows bearing engineered goldfish. And so I think that again, we're kind of slow in this first time getting to our first proof of concept in an aquatic invasive species. But over the years, it will become faster. We'll have new technologies available to work in parallel towards multiple different AIS targets.
Great. All right. I think we might have time for one last question. So I'm going to move on to this one. And it's asking, when deploying carp that produce minnow sperm, is there an efficient way to detect the few that weren't sterilized and still make carp sperm?
Uh, yeah, these would short answer is yes, because if we were doing this, carp are long-lived enough that you could, in theory, rear them in captivity in like a fish hatchery to the point where they're already sexually mature adults and test them, see what kind of sperm they produce. If there's that small number that are producing carp sperm, those would get put into a different bin and they would never be released into the lake for a control campaign.
All right. Perfect. That brings us to 159. So I'm going to use this last minute to give a couple of closing messages. Let's see if my slide wants to go. Here we go. Okay, so first, thank you, Mike, for joining us. We really appreciate you sharing all of this great new information about genetic biocontrol for invasive species. As I mentioned before, we did record this webinar. You can find it on our YouTube channel, which is z.umn.edu/AISTube. It takes us about a week to get things transcribed and up on there, but you should get an email notification once that is there and ready. You will also see a survey pop up when you leave, and we'd really appreciate it if you took some time to click that link and take our quick survey. I know there were just a couple of questions that we weren't able to get to in our time limits, but I do have Mike's email showing on the screen. So feel free to reach out if you have any lingering questions. And if you have questions about the webinar series or other questions about our AIS programs, you can reach us at AIS Detectors at umn.edu. I think with that, I'll bring it to a close. But thanks to everyone for joining us today, and thanks again for joining us and sharing all that you had, Mike.
My pleasure.